Today's topic discussion is cheese, cheese production. Previously, the class one of the unit two about the how the fermentations process run in the industrially, and there are the three fermentation steps. Next day, we discussed about the yogurt manufacturing details about the yogurt manufacturing steps, each and every details. And now today's class is all about the cheese production, cheese production process. Okay, cheese one of the fermented food. Cheese is one of the fermented food that start from simple raw material, simple raw material, and ends with huge diversity of color, taste, flavor, etc. So simple raw material means how the cheese production start. What is the raw material? It is the milk.
Some
It is the milk. So simple raw material. We are started from with the simple raw material, that is the milk, and we are ended with huge diversity of product, huge diversity of cheese. And today's we can differentiate the cheese on the base of color, moisture content, taste, and flavor. And now over 100 types of cheese are present worldwide. Over 100 types of cheese found all over the world. And cheese can be differentiated into based on color, taste, flavor, and the main different point is moisture content. Cheese classification based on moisture content. That means how much water present inside the cheese. And by that way, we can differentiate the cheese into hard cheese, semi-hard, and soft. In case of hard cheese, the moisture content will be very less. That's why the product is hard. After the manufacturing, it become hard. That is because of the low moisture content, and the low moisture content should be between 20 to 40% in case of hard cheese. And semi-hard cheese contain just more than 40%, 45 to 55 percentage. And the soft cheese, what will be the percentage? It should be the higher than the semi-hard and contain above 55, above 55 percentage, and goes to 60 or 65, etc. percentage. And some example of hard cheese, really hard because of the moisture content, is cheddar cheese. Example of hard cheese: cheddar cheese, parmesan. Example of some semi-hard: Limburg, Roquefort. And soft cheese example, cottage cheese, with relatively high moisture content, water content, camembert cheese, camembert, cheese classification based on the moisture content is three types. Clear? This is the initial part about the cheese. Now we are actually discuss about the manufacturing principle, manufacturing process and the steps of the manufacturing of cheese in industrial. So before manufacturing steps, we go through the principle of cheese production. How the cheese will made from a simple raw material and how the staggering diversity of the cheese will be achieved. Clear? So what is the principle behind the cheese production? So my next topic is principle of cheese production. It's very simple principle of cheese production, and you also can answer this. If you see the table of content, that means the raw basic material milk, what is the composition, and after the product, that is the cheese basic composition. Just check the composition: cow milk, raw material composition, and the after fermented product cheese composition. See the composition first, and you can answer what is the principle behind the cheese manufacturing. Okay, cow milk contain water, and the water percentage 87%. Fat percentage 4%. Protein 3.2%. Lactose, that is the principal sugar present in the milk disaccharide form, about 5%. And minerals less than 1%. Correct? This is the basic raw material cow milk composition. And let's look the cheese composition. After manufacturing product, what cheese composed? Cheese composed of water, and the water percentage 36 to 39%. It could be changed based on the hard, semi-hard, and soft, but the relative general cheese water content 36 to 39, below 40. Okay. Fat percentage 30 to 32%. Protein percentage 26 to 28%. Lactose less than 1%. Minerals 1%. And salt 2 to 2.5%. Just look the composition of milk and the cheese and check the what is the significance of the component difference. Check the guess the component difference. What is the significant difference between cow milk and the cheese? Answer. Am I audible to everyone? Answer. Where is the significant lies? What is the basic difference between the milk and the final product cheese?
Sir, more quantity of nutrients are present in the cheese as compared to milk.
Nutrients means which type of nutrients?
Proteins, fats, and as well as lactose is less in amount, so which
Less.
is good
So,
for pe-
okay, lactose is
people who are.
less. And you answered more nutrient. You cannot answer more nutrient. More nutrient is lactose is also is as a nutrient. It is a carbohydrate. So specifically increase in fat, specifically increase in protein, and decrease in water, decrease in water, not only the lactose, decrease in water and the lactose concentration. So these two are increased. Fat and protein are increased. In case of cheese, water is decreased in good quantity, as well as lactose also in a less quantity. So by looking at the composition of milk and cheese, what you can say? What is the principle behind the cheese production? হ্যাঁ এটা তো তোমার অ্যাডভান্টেজ হচ্ছে আমি বলছি যে প্রিন্সিপালটা কি মানে মিল্ক থেকে চিজ প্রোডাকশনের টাইমে জাস্ট কি করব হোয়াট ইজ দ্য বেসিক নিড ফর চিজ প্রোডাকশন ওকে ওকে জল কমালে আপনি ল্যাকটোজ কমে যাবে বিকজ ল্যাকটোজ ইজ কার্বোহাইড্রেট and carbohydrate means is a polar always present in the water and when water is removed lactose also be decreased. Okay, so the main things, the principle behind the cheese production is reduce the water, reduce
Lactose
the
is
water
decrease the
and
amount of.
okay, correct. Exactly. Reduce the water and increase the solid portion as protein and fat, not the carbohydrate. Increase the solid portion of the liquid of the milk as a protein and fat. And look at that.
No, sir.
That
Asking why lactose amount has decreased when we decrease the amount of water.
yes, because lactose is highly polar, and the water lactose is always present targeted to the polar. And when water is reduced, also lactose will be out from the product. Is it clear? Water is polar molecule and the lactose is polar. It's a carbohydrate, polar molecule. So polar always attached and that means when you remove the water, polar molecule lactose start to decrease.
Okay, sir.
Clear? So the cheese production basic mechanism is two things. From the composition, we can easily detect that the principle of cheese manufacturing based on number one, water portion is removed. Yes or no? Just remove the water. And number two, concentrate the solid. Concentrate the solids in the form of particularly mentioned in the form of fat and protein. So the principle behind the cheese manufacturing is based on these two steps. If you follow these two steps, ultimately raw material milk convert into the cheese. Yes or no? If you properly done these two steps, the cow milk, that is the milk, converted into cheese. So by keeping that way, we start to manufacturing, we enter into the manufacturing process. So clearly, cheese manufacture is based on simple by water reduced and concentrate the solid. How the water could be removed from the milk? So before manufacturing step, my question is how did the water can be removed? হবে, বাট চিজ প্রডিউস হবে না ওখানে, ওখানে থিক মিল্ক হয়ে যাবে, থিক স্লারি হয়ে যাবে, বাট চিজ ইজ দ্য কোয়াগুলেটেড ফর্ম and is a solid portion. So how did the water can removed from milk? Any guess? Monomer said about the heat treatment. Yes, heat treatment can reduce the water, but not the significant way. 80 from 87% to 36%, almost half, more than half actually. And second way to concentrate the solids. Remember that not only you reduce the water, also concentrate the solids.
স্যার দুধের মধ্যে মানে অ্যাসিটিক অ্যাসিড আর ভিনেগার এরকম কিছু দিয়ে
অ্যাসিটিক অ্যাসিড ভিনিগার দিলেই জাস্ট কার্ডলিং হবে, ওয়াটার থাকবে, বাট ওই কার্ডলিং এর মধ্যে ওয়াটার থাকবে, ওই কার্ডলিংটাকে আরো প্রপার করতে হবে, আর টাইট করতে হবে। ওটাতেও হবে, বাট কার্ডলিংটা ওটাতে কি হবে, সফট চিজ তৈরি হবে, যেটাকে আমরা বলছি কটেজ চিজ, দ্যাট মিনস অ্যাকচুয়ালি পনির, ছানা, ওটা সফট। বাট আমি যদি তোমার পার্মেজান তৈরি করতে যাই, দ্য ওয়াটার পার্সেন্টেজ ইজ মাচ লো, দেন কি করতে হবে, আরো টাইট করতে হবে কোয়াগুলেশন কার্ডলিংটাকে, কারেক্ট? So is for the cheese manufacturing, water remove steps utilize physical, chemical as well as biological events. So clearly water can be removed by, that is clear from our discussion, water can be removed by coagulation of milk, coagulation of milk, coagulation করলেই তো water বেরিয়ে যাবে, but water percentage still there in the coagulated form, curdling form, and we need the more solid, that's why more water should be released. So for cheese manufacturing, this coagulation done by physical, done by chemical step, as well as done by biological step. So three events makes the water reduce from the milk and make the more concentrated coagulate form, that is the cheese. And what is the physical form, chemical form and biological form? For the physical form, physical events, we are using heat and acid treatment, combination of heat and acid treatment. Okay, not only heat, also acid treated. Chemical means, what is the chemical events to reduce the water? Chemical events mean addition of acids that produce, not addition, produced of acid, it is not added, actually produced during the cheese manufacturing step, produced acids by lab. Produce acids by lab. Lab means lactic acid bacteria. So in the starter culture, what we are going to use, which bacterial strain? We are using particular lactic acid bacteria, and make the starter culture and use in the cheese production. And the biological steps, we are using chymosin. Chymosin. It is an enzyme. It is an enzyme. So we are combining three events: physical, chemical, and biological. And these three events make the milk more coagulated, more solid form. The physical means heat treatment, specifically heat treatment, and some kinds of acid treatment also be done. And the chemical treatment, actually the starter culture that is produced during the formation of cheese, that is the acid, lactic acid. And the biological is the using of enzyme. So here enzymes also added during the manufacturing steps. A step is there where the particular enzyme is given, and these enzymes curdle the more, curdle the milk more and reduce the water. Clear? Because we need to reduce the water more significantly, more than double. Cheese content 36 and water content in the milk is 87. So huge quantity of water should be released, and this is not a single step process, and it should be combined effort of physical, chemical, and biological. Clear? Now let's look the manufacturing steps of the cheese industrial. General steps of cheese production. First event, milk standardization. Milk, basic raw material, milk should be standardized before fermentation. Most cheeses are made from cow milk. So the starting raw material could be differ based on the cheese type, but most of the time we are using cow milk. But the preference should be given whenever the different type, unique type of cheese should be produced, like feta cheese. Feta cheese is done by using goat milk. Mozzarella, common to everyone. Mozzarella cheese. This cheese is produced from the milk that is taken for mozzarella is water buffalo milk. বিভিন্ন প্রাণীর সেক্ষেত্রে তো বিভিন্ন ধরনের কম্পোজিশন আলাদা হবে হ্যাঁ goat milk water buffalo milk composition different and based on that the texture flavor also be changed so the unique type of cheese can be produced from different types of source of the milk but the most common user is the cow milk. Roquefort cheese, for that we take sheep milk. Sheep milk. So first standardize the milk. Standardize in the standardized state. First you have to choose which type of cheese you are produced, and based on that choose the basic raw material. Clear? If you are produced a mozzarella, then start with buffalo milk, not the cow milk. Correct? So based on the cheese type, the raw material can be changed. That is the first point of the milk standardization. Second point of milk standardization, milk should be pasteurized or can be used as a raw, can be pasteurized or used as raw. That means unpasteurized milk also can be used, but in case of yogurt manufacturing step, we always produce pasteurized, and actually above the pasteurization temperatures are used. Not that pasteurized temperature, above the pasteurization temperature used for a special benefit, but here raw milk can be used. And many countries, mainly European countries, they are using for the cheese manufacturing raw milk. But strictly in the US, they are produced from pasteurized milk because pasteurized milk has advantage as well as disadvantage. What is the advantage? Same advantage that are used in the yogurt manufacturing. That means it kills major foodborne disease causing microbes. It kills spoilage causing microbes. That's why the milk's chance of milk spoilage, foodborne disease would be less, incident would be less. But the disadvantage is during the pasteurization process, the high treatment, high temperature treatment change the flavor, change the taste of the cheese that we get from the pure milk that is not formed as a pasteurized milk. Okay, so pasteurized milk used in US country, USA to make cheese, and they are using because uh, for low incidence of spoilage and foodborne disease. Because pasteurized temperature actually kill that type of microbes. By that way, it is safe. But pure or unpasteurized raw milk can be used in some countries, like European countries mainly, and that is because of the exact flavor, exact taste. So both way we can start milk cheese production. We can start in pasteurized way, pasteurized milk. We can take pasteurized milk or unpasteurized milk. We can also we can use for cheese manufacturing. Milk standardization process, clear or not? Understood, everybody?
Yes, sir.
After milk standardization, the next step is addition of starter culture. Step number 2. Okay, in India, pasteurized milk are used. Okay, we are following the guideline of USA. That's why we are used pasteurized milk for the cheese manufacturing. Okay, next step number 2, addition of starter culture. And clearly, the starter culture that we are going to use is lactic acid bacteria. And lab has different strain, different species. So the species that you choose based on the cheese manufactured types of cheese. So point number two, the types of lab depends on the intention of cheese maker. And how does it influence? Based on the cheese maker process intention, the lab will be differ based on intention of cheese maker, because some cheese production step needs high temperature, okay, and some cheese production step need low temperature, and based on that the starter culture should vary, the lab species should vary, because that high temperature needs high resistance at that temperature. For that we should select those microbes which can grow at high temperature, yes or no. But the low temperature process needs not the thermophilic strain, it needs normal mesophilic bacteria. Thermophilic mesophilic bacteria means mesophilic bacteria that grow at the room temperature, that means 30 to 40 degrees centigrade at better way. And the thermophilic bacteria, those bacteria which grow at high temperature, they are actually love to grow at high temperature. That means above 50, 60 degrees. So based on the cheese manufacturing steps, the starter culture, the lab species also vary. Thermophilic LAB lab can use, as well as mesophilic lab lactic acid bacteria can be used. Thermophilic lab example Streptococcus thermophilus, Thermophilus. These 10 species also used in the yogurt manufacturing step. Streptococcus thermophilus. Lactobacillus velvetians. These two strain can be used when the manufacturing step need more temperature, high temperature, because these lab are thermophilic and they can easily survive at high temperature and can grow. Correct? But some cheese manufacturing step needs moderate temperature. That means normal room temperature or just above the room temperature, 40-45 degree. That's time we do not need thermophilic. That time we need mesophilic lab. And some example of mesophilic lab that we are used during the cheese manufacture: Lactobacillus lactis, Lactobacillus cremoris. So question can become with the statement question: during cheese manufacturing process, the starter culture lab can be varied based on type of cheese produced. Statement is correct or false? Justify. Statement is correct or false? During cheese manufacturing process, the starter culture vary based on the types of cheese production. Statement is correct or false? Statement is 100% correct. The type of cheese can be changed the type of starter culture. The cheese manufacturing state that needs more temperature, that time we are going to use thermophilic lab. And the example of this Streptococcus thermophilus and Lactobacillus bulbitans, or that time when the cheese manufacturing needs a moderate temperature, we are going to use mesophilic rather than thermophilic, because mesophilic bacteria are grow better at above the room temperature, just above the room temperature. And that time we are going to use Lactobacillus, lactis and primordial. Clear? Answer is clear. Starter culture point one done. Understood? Starter culture point one done. Addition of starter culture point one done. That means starter culture can be varied. Okay. And the point number two, we also know that lab can be of two types. Lactic acid fermentation can be of two types, and based on that, lab are two types. One is homolactic lab, other is heterolactic lab. Yes or no? Homolactic lab, and heterolactic lab. So, what type of lab we are used in the cheese making, or heterolactic, or both? Homo or hetero or both? We are using both, not homo. So not only the our target is to produce lactic acid, other products that also be produced inside the cheese, and that make the cheese more flavored, more tastier than the that we are using only the homolactic or heterolactic. If we use only homolactic or heterolactic, the cheese flavor is not certain amount, but both, when you use both, that time the cheese is more flavorful. So the point number two is একদম এটাই আমাদের টাস্ক। কারণ দেখো অর্গানোল্যাকটিক মানে হচ্ছে তোমার ফ্লেভার টেস্ট, if it will be enhanced, that will attract more customer, more buyer, right? So that is the main purpose of food manufacturing step. Then the point number two, we are going to use both type of lab, both homolactic and heterolactic LAB. That means not only the lactic acid, because homolactic process produce lactic acid. That's clear, very clear. But heterolactic process produce lactic acid as well as other product. Yes or no? Ethanol, acetyl CoA, other acids, etc. And that make the product is more flavored, more tasty. Correct. That's why we are going to use both type of lab, homo and heterolactic lab, and this type of microbes labs, Lactobacillus lactis, Primordial, Belvitans, and the Thermophilus, all are homolactic. All are homolactic. That's why we further added some heterolactic lab like leuconostoc. For that, we further add heterolactic lab like leuconostoc. So starter culture composition of cheese making, it is clearly lab. Starter culture composition of cheese making, it is clearly lab, and lab can be used in the both types of lab, that means homo and heterolactic, as well as the based on the cheese making, the lab is also different. Starter culture is done. Step number 2 is done. If any question you can ask. আমরা এখানে অ্যাক্টিভ ল্যাঙ্গুয়েজ করছি যাতে আমাদের অর্গানিক প্রপার্টি আরো বাড়ে আর জেনারেল যুক্তরাষ্ট্রে অ্যাড করে দিচ্ছি because these all are homolactic. Previous examples all are homolactic. So further we added some quantity of some concentration of leuconostoc in the starter culture to add more heterolactic organoleptic property. Taste, texture, flavor, etc. Done. Starter culture done. Step number 3. Step number 3, coagulation or curdling. Step number 3, coagulation or curdling. For that, chymosin is an enzyme. Okay, chymosin is added to the milk. Immediately after or after pre-ripening state. Pre-ripening period mean, what is pre-ripening period? Pre-ripening period mean the time for starter culture to grow and establish into the milk. That time is called pre-ripening period. So what is pre-ripening period? The period or the time where starter culture grow and establish in high quantity inside the milk. So we can add chymosin, that is an enzyme. This is an enzyme, chymosin, that can be added just after the addition of starter culture or need pre-ripening period. After that, we can add the chymosin. So what do you think? Which is the better time to addition? Just after the starter culture addition or maintain the pre-ripening period? After that, you add the chymosin. Which is the better time to add chymosin in the milk? Maintaining the pre-ripening period. Other, try to answer other online student.
Pre-ripening period, after the pre-ripening.
After the pre-ripening period. Anyone other? What is the better time? Just immediate after the starter culture addition or maintain the pre-ripening period, then use starter culture, then use chymosin. Monomer, pre-ripening period. So majority answer is pre-ripening period, and the answer is correct. But why? Why pre-ripening period is better, not the just immediate after the starter culture? এক্ষেত্রে স্টার্টার কাজে একটু সময় পেয়ে যাবে, যে সময়টাও মানে ইউজারের মধ্যে চিজের ফরমেশন টাইম পেয়ে যাবে। চিজ ফরমেশন এখনো হয়নি না, জাস্ট স্টার্ট হয়েছে, স্টেপ নাম্বার আমরা থ্রিতে আছি। আমরা কার্ডলিং করাচ্ছি মানে কোয়াগুলেট করাচ্ছি, সলিড গুলোকে কোয়াগুলেট করাচ্ছি, দ্যাট মিন্স প্রোটিন ফ্যাটকে কোয়াগুলেট করাচ্ছি। এবার আমরা জাস্ট বলেছি যে প্রি রাইপেনিং পিরিয়ড মেনটেন ইজ বেটার, আফটার দ্যাট কাইমোসিন অ্যাডেড, that works better after pre-ripening period. Why? Okay, I give you another hint. Chymosin, it is an enzyme and it is a acid protease. Which type of enzyme it is? Acid protease. Now you can answer why pre-ripening period maintenance is better for chymosin to what? And it will be a question I write after the discussion.
Sir, because the lactic acid bacteria will produce lactic acid and after that the chymosin enzyme will act.
After that, chymosin enzyme act better because it is a acid protease. We know enzymes need particular pH to activate. Particular pH to activate, and this enzyme is acid protease. That means it needs acidic pH. It needs acidic pH to activate more to work better. So chymosin is an acid protease and it needs lower pH to work. So now see, if after just addition of starter culture, can you achieve the lower pH in the milk? No, pH so originally is a alkaline, that means above 7. So just after addition of starter culture, chymosin is activated or not? Chymosin is less activated. Chymosin can work, but it is not optimal as work at lower pH. But when we maintain the pre-ripening period, that means we are giving the time for starter culture to grow, and starter culture contain lab, that means produce lactic acid. Yes or no? So pre-ripening period, during the pre-ripening period, starter culture produce lactic acid. Yes or no? Lactic acid, and this lactic acid make the overall environment low pH, and this low pH activate or works, that low pH works chymosin better. Clear? So that is the potential question, and the question is why free ripening period is better for cheese production, better for coagulation. Answer is clear. Now the source of chymosin. Where does the chymosin can we get? Source of chymosin. Chymosin is naturally found in the ruminant animal in their stomach to curdle the milk in that animals. So chymosin naturally we can found in the calf, or we can engineer microbes that produce chymosin. And for that we need recombinant DNA technology or genetic engineering. That is another separate subject. So source of chymosin naturally we can found in calf stomach, ruminant animal calf stomach, but for that you have to be slaughtered the calf. So one issue. So we can circumvent that issue by means of producing microbes, engineered microbes that producing chymosin. So no problem in that scenario. Microbes are modified. No microbes are produced chymosin. Now microbes are modified by genetic engineering, that is separate subject, recombinant DNA technology, and now microbes start to produce chymosin. But chymosin is 100% needed in the cheese making. So in worldwide, huge quantity of cheese is produced every day. So if we are isolating chymosin from the calf, huge quantity of slaughter should be done. So that is not permissible, right? That's why we are start to produce microbes, that engineered microbes that produce chymosin. So chymosin is the acid protease that create the curdling and coagulation of the solid. So point number 2 after this, chymosin create curdling. Curdling means simple language lump formation, coagulate. Step number 3 clear. What is chymosin? What is the source of chymosin? Important question. And what is the use of chymosin in the cheese making? Important question. Step number 4. After coagulation or curdling, the step number 4 is cooking and cutting. Cooking means at higher temperature, some type of incubation is done. So first point, after coagulation, cut into the section, cut the coagulate mass into the section, as well as after the sectioning, it should be cooked at low temperature, or some cheese need high temperature, and that is the step. Based on that, the lab is being chosen. When low temperature is used for the cooking, we are using mesophilic lab. When high temperature is used, that time we are using thermophilic lab. So that is the step previously we discussed. For that, we change the lab species. Okay, it should be cooked at low temperature or high temperature. So what is the need for that? What is the need for cutting into the sectioning the coagulated mass, and after that cooking? What is the need? What is the function of cutting and cooking? Anyone? হ্যাঁ, চিজ তো ফর্ম হবে। চিজ ফর্ম করতে গেলে দুটো প্রিন্সিপাল একটু মাথায় রাখো আবার। একদম ব্যাকে যাও। ফার্স্ট টাইমে কি বলেছিলাম? ওয়াটার শুড বি রিমুভ মোর অ্যান্ড মোর অ্যান্ড মোর। দ্যাট টাইম কি লাগবে? আমি যদি কোয়াগুলেট মাছটাকে সেকশনিং করি, the surface area will be more now and the water release chance of water release more. যদি আমরা একটা কোয়াগুলেট মাছকে আমরা যদি জাস্ট কুকিং এ বসিয়ে দিই, that water is less removed because it holds more water inside. So after sectioning, cutting into the section, the surface area becomes high and the more water can be reduced. Number one. And the cooking step followed by the cooking step for remove the water. So this step is needed for cheese making. What is the function of cooking and cutting? This step, or what is the importance of this step? Because it induce or it enhance syneresis. It enhance, in food microbiology a term is called syneresis. Syneresis means release more water, releasing water. So for that, cutting and cooking is needed. Cutting into section, increase the surface area, and more water can be removed. That is our purpose, to remove more and water. So surface area, increase the surface area, water will automatically remove. As well as when we cook, that temperature also induce water to release. Clear? For that, cooking and cutting step is very important in cheese making. Otherwise, we cannot make hard, semi-hard cheese. Understood my point? Everyone, online students, answer.
Yes, sir.
Yes, sir.
Step number 5. Step number 5, another important step for reducing the water as well as other benefit is addition of salt. Addition of salt. Check the cheese composition. In case of milk, there is no salt, but cheese contain 2 to 3% of salt. Why? Because in the manufacturing step, we deliberately use salt, addition of salt in making of cheese. Why? Importance of salt. So স্টেপ নাম্বার 6 অবশ্যই টেস্ট এনহান্স করে, আরো প্রপার্টি আছে, আসছি। স্টেপ নাম্বার 6, সল্ট অ্যাডিশন, সল্টিং। হুম হুম ও এটা লিখবে, ও 5 হবে, কারেক্ট। সল্টিং, salting is essential ingredients for cheese making. And why it is essential? Why it is essential? Because number one, salting increase the flavors, enhance flavor. Obviously, without salt, it tastes like a bland, right? So enhance the flavor. Number one. Number two, also enhance the synergy. Enhance the synergy means water released from the curd. Water released from the curd. So addition of salt also enhance synergy and curd will be now become more hard. Yes or no? And why synergy occur after addition of salt? Why synergy occur? It is a osmosis, clearly osmosis process and exosmosis process. Exosmosis process and make the water out from the solid curd. So enhance the synergy by means of clear cut. The phenomena is here utilized is osmosis, because when the salt is outside more, water is less, and inside the card water is high. So water is coming from the high concentration to low concentration. So pure osmosis occurs when you add something, add salt into any kind of. You also see in the vegetables also. In case of cucumber also, add some salt, it will be more moisture, because addition of salt create the osmosis process. Yes. Number three, salt also enhance the preservation time, induce preservation of cheese. How? How? Because many microbes by addition of salt are being killed, are being killed by addition of salt. How it preserved the cheese in the longer period? Because it killed the spoilage causing organism. At high salt, microbes are not being healthier environment for growth. That's why salt addition make the preservation of cheese more. Now you can see why European people without pasteurized milk can start the cheese making process. Because at the time of salting, many microbes are killed. No pasteurization is absolutely necessary for cheese making. Clear? The salting step also kill the microbes. That's why European manufacturing step they are not used pasteurized milk. But for further safety, we start pasteurized milk. We start with the pasteurized milk for further safety purpose. But absolutely not needed. That step is absolutely needed for pasteurized milk should be needed for the cheese manufacturing. That is the not scenario, because salt can kill many microbes. Clear? And induce the preservation time. After salting, the step number 6 and the last step is ripening and or aging. Ripening means actually aging. Shelf life বাড়ানো, taste, flavor change করা, কারণ after that cheese that you produce, the taste after ripening, it will be far different. So cheese ripening and aging done in the particular temperature and involved in a particular separate room from the manufacturing room. So ripening is important for final quality and properties of cheese. And during the ripening period, various enzymes and microbes slowly act on the cheese and change the taste, change the flavor continuously when you give the time of ripening. So various enzymes and microbes present in the cheese are responsible for ripening, and these microbes enzymes can come from the indigenous, not indigenous, from the product itself, because we add starter culture, we add enzyme previously. So these enzymes, these microbes continuously do their work and change the cheese flavor, taste, etc. So these enzyme microbes are actually product, came from the product, or we can further add some enzyme, we can further add some microbes that enhance the taste in the ripening, or can be added. Penicillium, that is a fungus mold, can be added at that time, ripening time, to create more flavor, texture, color, etc. So ripening step actually create the final outcome of the cheese. Clear? And that vary, ripening and aging step vary the cheese type, cheese different texture and flavor type. হুম 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 হ্যাঁ, হোলগুলো তো আগেই হচ্ছে রাইপেনিং স্টেপে আরো এটা ইনক্রিজ করবে, যেটা সুইস চিজ, কার্বন ডাই অক্সাইড রিলিজ। So aging purpose vary, ripening period vary. Some need 4 to 6 weeks. Some cheese need 6 to 10 weeks, and some need 14 to 18 weeks. And based on that ripening period, the name also be differed. And 4 to 6 weeks ripening period cheese are called young cheese because it needs less ripening period, less ripening time. 6 to 10 weeks, it called mid mature cheese, and 14 to 18 weeks called extra mature cheese. Obviously, extra mature cheese, that means the cheese need more ripening period. The cost will be more, and the taste and flavor it way different from the young cheese. Yes or no? So ripening period and aging period. After that, the cheese is manufactured. Any question from cheese manufacturing process? Every steps I discussed thoroughly, details. If any problem, you can ask now. হ্যাঁ বলো।
সেকেন্ড স্টেপে যে এনজাইমটা আছে সেটা মাইক্রোবসেরই প্রোডাক্ট, তাই তো?
আগে তো কাপ থেকে নেওয়া হতো, কাপ ইন্টেস্টাইন থেকে নেওয়া হতো রুমিনেন্ট অ্যানিমেলের। এখন এটাকে ইঞ্জিনিয়ার দিয়েও প্রডিউস, এত হিউজ প্রোডাকশন নিডস হিউজ কোয়ান্টিটি অফ এনজাইম, সো এটা পসিবল না স্লটার্ড কাপ থেকে নেওয়া আইসোলেট করা। দ্যাটস ওয়াই আমরা এখন চেঞ্জ করে ম্যানুফ্যাকচার করছি ইন ব্যাকটেরিয়া। So two principle is achieved after six step. Two principle that we discussed previously at the time of cheese manufacturing. One is reduce the water and second is concentrate the solid. So to reducing the water, what are the steps are involved? Lactic acid bacteria create the lactic acid production. That acid also coagulate, and that acid production induce the chymosin to react more, and chymosin actually lumping coagulate curdling second time, and third time when we add cooking temperature and the sectioning, that means cutting, the more water is released. After salt addition, another round of water is released, and by that way the coagulated mass is very hard and shape as you wish, and make produce a cheese block or something. একটা চিজ ব্লক হেভি হার্ড হয় না, ওটাকে তো তোমার যখন ইউজ করা হয় ওটাকে ঘষে গ্রেট করে দেওয়া হয়, তার মানে ভেরি হার্ড হয়। এত থিক হয় কি করে? তিনটে স্টেপে ওয়াটার রিডিউস হতে 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 চিজটাকে সলিড থিক বানিয়ে দিয়েছে। ক্লিয়ার? চিজ ম্যানুফ্যাকচারিং কিন্তু ডিফিকাল্ট, হ্যাঁ, ম্যাক্সিমাম স্টুডেন্ট বোঝে না। বাট আমি জিজ্ঞাসা করছি ফার্দার, ইফ এনি কোশ্চেন আফটার মাই পড়ানোর পরে, ইফ এনি কোশ্চেন ইফ হ্যাভ ইউ ক্যান আস্ক। মিল্ক থেকে দুটো প্রোডাক্ট হেভিলি ইম্পর্টেন্ট এই ম্যানুফ্যাকচারিং এ। ওয়ান ইজ ইয়োগার্ট ম্যানুফ্যাকচারিং, যেটা ডিটেলস থেকে কোশ্চেন করে, অ্যান্ড সেকেন্ড ওয়ান ইজ চিজ ম্যানুফ্যাকচারিং। এই ডিটেলস এর স্টেপ গুলো থেকে ভেতর 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 থেকে কোশ্চেন তোমাকে করবে। ইফ ইউ আর নট ক্লিয়ার ইন এভরি স্টেপ, ইউ ক্যান নট আনসার। আর তোমাকে যদি আমাকে এরকম একটা জাস্টিফাই মার্কা কোশ্চেন তুলে দেয়, তুমি শুধু স্টেপ মুখস্থ করে চলে গেছো, তুমি আনসার করতে পারবে না, আমি 100% শিওর। বিকজ তোমাকে এভরি কর্নার তোমাকে জানতে হবে যে কোথায় কি হচ্ছে, কেন হচ্ছে, কেন দিচ্ছি আমরা। ঠিক আছে? যে তুমি শুধু পড়ে গেছো কাইমোসিনের, কাইমোসিনটা কি, কোথায় পাওয়া যায়, আরে কাইমোসিনটা কিভাবে ইউজ হচ্ছে, প্রিন্সিপালটা কি, কাইমোসিন কিভাবে কাজ করছে, সেটা তো তোমাকে জানতে হবে। আর কোশ্চেন তোমাকে যে জায়গা তুমি পড়বে না ঠিক ভাববে, সেই জায়গা থেকে কোশ্চেন দেবে। যে জায়গাটা তুমি ছেড়ে গেলে এবং সেই কোশ্চেন এবং ক্যালকাটা ইউনিভার্সিটি সেই জায়গা থেকেই কোশ্চেনটা তোমাকে তুলে দেবে। আর তুমি ওখানে মানে হেড স্ক্র্যাচ করবে, এদিক ওদিক তাকাবে, আলটিমেটলি কোনো লাভ নেই, কারণ তুমি জায়গাটা ছেড়ে এসছো।
I have a question.
ইয়েস।
Sir, in the ripening step, various
Yes.
enzymes and microbes slowly act on cheese and change the flavor. So when we added salt in the previous steps, so microbes got eliminated. So then how come these
Not
microbes
all
are left?
microbes, not all microbes. I said clearly the spoilage causing microbes, those are not surviving in the high salt. Those microbes are killed, but lactobacillus, those starter culture microbes, that microbes are easily survived. And by that way, the starter culture is produced. Starter culture means key is a particular formulation of microbes that are being used at manufacturing steps without any disturb. So after salting, the microbes in the starter culture can grow.
এবার একটা প্রশ্ন আছে,
Okay,
সল্ট
sir.
ইউজ করেছে ওরা বয়েলেজকে কমানোর জন্য, সল্ট তো ততটাও সেফ না যতটা পাস্টুরাইজিং সেফ।
অবশ্যই, বাট সল্ট শুধুমাত্র স্পয়েলেজ কমাচ্ছে না, তার সঙ্গে আরো কিছু অ্যাডিশন করছে। ওয়াটারকে ফার্দার রিলিজ করাচ্ছে এক্সোঅসমোসিস করিয়ে, ফ্লেভার ইনডিউস করাচ্ছে, নাহলে তো ব্ল্যান্ড টেস্ট আসতো। ফ্লেভার ইনডিউস করাচ্ছে। সো তিনটে পারপাসে সল্ট ইউজ হয়। তো সল্টেরও ফাংশন চায়। ওয়াই সল্ট ইজ অ্যাডেড নট ইন দ্য ইয়োগার্ট বাট ইন দ্য চিজ, ওয়াই? বিকজ অফ দ্য পারপাস, থ্রি পারপাস। ঠিক আছে? হ্যাঁ, এবার তোমার কোশ্চেন কি পাস্তুরাইজ মিল্ক করলে আরো বেটার? দ্যাটস ওয়াই ইউএসএ কান্ট্রিতে অলওয়েজ পাস্তুরাইজ দিয়ে স্টার্ট করা হয় যাতে বেটার সেফটি পাওয়া যায়। হ্যাঁ, তবে তুমি যে পিওর যে তোমার এই কেসটা ওটা কিন্তু চেঞ্জ হবে। তুমি পাস্তুরাইজ সেম একটা তুমি পিওর মিল্ক দিয়ে র মিল্ক দিয়ে বানাবে, টেস্ট ইজ ডিফারেন্ট, টেস্ট ইজ ডিফারেন্ট ইন টু প্রোডাক্ট। ঠিক আছে। আর চিজের অরিজিনেটর হচ্ছে মেনলি ইউরোপিয়ান কান্ট্রি, তো ওরা বেসিক্যালি ইউজ করে, প্রেফার করে র মিল্ককে ইউজ করে চিজ বানাতে। আর খুব কিছু প্রবলেম হয় না, তার কারণ হচ্ছে সল্টিং স্টেপ আছে, তারপরে হচ্ছে আমরা these two steps actually eliminate many spoilage organism as well as disease causing organism. But for further safety, better safety, we are now used pasteurized milk. In India country also, pasteurized milk is the starting material. হ্যাঁ 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 হ্যাঁ, আমুল টামুল তুমি যাই খাও না কেন সবই পাস্তুরাইজ মিল্ক দিয়ে করা হয়, and after pasteurization it is started. Any other question?
Sir, after this cheese production, how much
Yes.
portion will be left in unit 2?
How much portion? Other fermented products also there, acidophilus milk, buttermilk, as well as kefir, or 3 or 4 products. And these are all small part. 2 major parts is completed. One is yogurt, other is cheese. Because various intermediate steps questions can be given. Other milk fermentation steps are very easy, and just we are started with we should know the starter culture. What is the starter culture is used? What is the raw material? And what is the basic steps? That's all. That's all. But these steps, these two fermentation product very critical. That's why details se discuss করা হলো।